We have identified our problem, whatever that may be. We have formed a hypothesis, which means we have a possible explanation, a potential explanation. Now we need to test that explanation. So we need to do something like either experiments in the lab or maybe make more observations, something in order to test that hypothesis to see if it is in fact supported. Is it a good explanation or should we toss that explanation out? So that's the point we're at right now. So in order to do that, got to keep a few things in mind. In experiments, or actually you're just observing the outside world, we have what are called variables, and that's where what we're gonna collect data on. A variable. A variable is anything that varies. So, for instance, not everyone at Slick is the same height. There's differences in height, so that varies. What else is different? Not everyone's the same age. A lot of different things vary. So really, so many things in this world vary. You really need to hone in on exactly what you're trying to know. What are you trying? What question are you trying to answer? What hypothesis have you offered? And you really narrow it down to what variables you're actually going to collect data on. And there's two main types of variables, and I always get these confused because independent and dependent. Well, you'll see. They seem counterintuitive to me. The independent variable is the thing that the researcher, let's say you, can actually control. Changes, or I would add, control. So in this particular experiment, we're testing some fertilizer to see how well it helps plants grow. So I can control how much fertilizer I dump onto this plant. I could dump a lot onto some plants. I could dump none on other plants and see what happens. I can control how much fertilizer. So I can control that. It's not the same as a control group, which some of you might have heard. Let's just say I have control over it as a researcher. Not the same as a control group, which we'll get to in a minute. Some of you may have heard of that. So how much of something, anything can vary. Amount of fertilizer can vary. I can control how much. That is the independent variable. The reason that's counterintuitive to me anyway is if I can control it, I feel like it should be dependent on me, and that the independent thing should be the thing that's independent of me and I can't control. It's not. Sorry. The dependent variable is the variable that hopefully maybe is influenced by the independent variable. If my hypothesis is true, the independent variable will determine the dependent variable. So in this case, it would be like, how much does the plant grow? How many leaves does it have? How many fruits does it make? Whatever the case may be. This is what I'm watching to see if it changes as I change my independent variable. So these are things that the researcher cannot control directly. They're hoping to control via, the independent, but they're not sure. They're not sure if the independent's going to affect the dependent. If it does, then great, my hypothesis is supported. If it doesn't, my hypothesis rejected, toss it out the window. So this is something you can't control directly. That's your dependent variable. Okay. Great. One other, or actually two other, scientific concepts I'm going to go over here. Sample size. This is absolutely key. In science, we always talk about, well, what was the sample size? So the larger the sample size, good experiment. Let's say I wanted to know the average height of students at Slick. If I measured 2 people, that's not much of a sample size. You need to measure a lot more. I don't have time to measure everyone at Slick. There's like 60,000 plus students. But if I got, a pretty big sample, that's why we call it a sample. We don't have time to measure the entire population. So maybe I could measure a couple hundred sample at Slick, and that would be a good experiment. A small sample size of like 2, that's a bad, poor experimental design. It's not going to be scientifically valid. You didn't measure enough people. What if you happen to measure the two shortest people, or two just particularly short people, or two particularly tall people? You're not going to get a sense of the average height it's like. In this silly cartoon, the Dogbert Research Company. Have you ever been killed by a poorly designed product? Now I'm hoping you recognize the fallacy there. You can't ask somebody if they've ever been killed, because presumably, at least according to our current scientific knowledge, you can't talk to the dead. Again, current scientific knowledge. It is very tainted research. It's terrible. Shows that products haven't killed anyone. But for an extra $50,000, you can ask a second person. I want this consulting job because, man, I'd love to make one phone call and make $50,000. Wouldn't we all? But the point here being, you can't just ask one person. You can't do that. So in the case of our pandemic, you can't just measure one person's, symptoms and say, well, that's going to be the symptoms for coronavirus. You've got to look at a lot of people, a large sample size, to see what is the actual symptoms that you expect to see, because it could be a range. You need a big sample. Okay, I think I'm going to leave it at that. Sample size and independent, dependent variable.